welcome back to my channel. In previous video, I just discussed about the basic immunity, innate and adaptive immunity. Now I am going to discuss about the immunological memory and vaccine. What is immunological memory? When B and T cells are activated and begin to produce the duplicate cells, some of their offspring will become long-lived memory cell. This memory cell activate further B cell and T cell against the recurrent repetitive antigen. Throughout the lifetime of an animal, these memory cells will remember each specific pathogen encountered and can mount a strong response if the pathogen is detected again. This is adaptive because it occurs during the lifetime of an individual as an adaptive to infection with that pathogen and prepare the immune system for further challenges. Immunological memory is ability to remember foreign substance previously encountered and react again promptly if the pathogen is detected again. There are two kinds of immunological memory: passive short-term memory, active long-term memory. Coming to the first, passive short-term memory. In the passive immunity, antibodies come from outside the person's body. Passive immunity is usually short-term, lasting from few days up to several months. Several layers of passive protection are provided by mother. In pregnancy, for example, the antibody G is transported from mother to baby. It directly crosses the placenta. So, human babies have a high level of antibodies even at the birth. And next one is breast milk. It also contains antibodies that are transferred to gut of the infant that protect against the bacterial infection until the newborn can synthesize their own antibodies. Going to say about the active long-term memory. Long-term active memory is acquired following infection by activation of B cell and T cell. When a clone of T lymphocytes is activated by an antigen, many of the newly formed lymphocytes are preserved in lymphoid tissue to become additional T lymphocytes of that specific clone, which are called memory cells. Can you understand? During activation of the T lymphocytes, and some of the lymphocytes are preserved in lymphoid tissue, that is called memory cells. These memory cells even spread throughout the lymphoid tissue of the entire body. On subsequent exposure to the same antigen anywhere in the body, release of activated T cells occurs far more rapidly and much more powerful than had a first exposure. Thus, the new lymphocytes are added to the original lymphocyte of the same clone. They also circulate throughout the body to all lymphoid tissue. However, they remain dormant until activated once again by the new quantity of the same antigen. These lymphocytes are called memory cells. Subsequent exposure to the same antigen will cause a much rapid and much more potent antibody response in the second time. Coming to the immune system and vaccine. A host organism needs time, often days, to mount an immune response against a new antigen. But memory cells permit a rapid response to pathogens previously encountered. The active immunity can be generated artificially through vaccination. Okay, active immunity is generated artificially through the vaccination. The principle behind the vaccination is to introduce an antigen from pathogen. That means introduce an antigen from the pathogen in order to stimulate the immune system and develop specific immunity against that particular pathogen without causing disease associated with that organism. Here, the pathogen is maybe bacteria, virus, fungus, but introduce antigen means there is part of pathogen. It may be a glycoprotein, protein. The active component of the vaccine that is responsible to protect against particular viral bacterial infection can often consist of many forms. Killed or inactivated vaccine contain pathogens that have been returned harmless by treatment with chemical or high heat. Killed vaccine or inactivated vaccine is the pathogen is treated with chemical or high heat. Live attenuated or weak vaccines contain live pathogens that have accumulated mutation so that they are no longer virulent to human cells. These vaccines are almost commonly generated by repeat infection of the pathogen in culture cells until its virulence is lost. Subunit vaccines contain a purified protein component of the pathogen. Such protein can be either isolated from infected material, such as a blood from chronically infected patient, or generated by recombinant methods. Coming to toxoid vaccine. Toxin produced by certain bacteria, example tetanus or diphtheria, are used against pathogens that employ an extracellular toxin to cause disease. This vaccine contains a form of toxin that has been inactivated by treatment with chemical or high heat. Toxoid vaccine are toxin is produced by the tetanus and diphtheria, but this toxin is inactivated by the treatment with chemical or high heat. The treated chemical and or high heat toxin is introduced in our body. Coming to primary and secondary response. The formation of antibodies that occur on first exposure to specific antigen is called primary response. The formation of antibodies that occurs after second exposure to the same antigen is called secondary response. The potency of primary response is weak and short. The secondary response, by contrast, begins rapidly after exposure to antigen, is far more potent and form antibodies for many months rather than the only few weeks. Here you can see primary response. First attack from the antigen, immediate action, rapid, and the duration is low comparatively second response. Okay, the production of the antibody concentration is less in primary response, but the secondary response is high. And coming to now histocompatibility molecules. Histocompatibility molecules originally identified as antigen that stimulate rejection of transplanted organs, which are now known to be extremely important for induction and regulation of the immune response. These are present on the surface of the antigen present in cells. During transplantation organs, we should investigate about the histocompatibility molecules. Physiologic function of the cell surface histocompatibility molecule is to bind peptide fragments of foreign protein for presentation to antigen specific T cells. In human, histocompatibility proteins are encoded by a large group of genes that is called major histocompatibility complex or human leukocyte antigen. So named because MHC encoded antigen were initially detected on leukocytes. In human, the major histocompatibility proteins are human leukocyte antigen on the basis of their chemical structure, tissue distribution and function. This is classified into three categories: class 1, class 2, class 3. Here, class 1 proteins present in all nucleated cells and platelets. Class 2 proteins present in the B cells, macrophage, epithelial cells, activated T cells and dendritic cells. Class 3 proteins present in the components of the complement system and some of the cytokines. Coming to disorder of human immunity. Failure of host defense leads to disorders in the human immunity. First, it may be immunodeficiency and autoimmunity, and finally hypersensitivity reactions. First, immunodeficiency. Immunodeficiency occurs when one or more component of the immune system is inactive. In developed countries, obesity, alcoholism, and drug use are more common cause of poor immune function. However, malnutrition is the most common cause of immunodeficiency. And autoimmunity. Failure to differentiate host cell from foreign antigen can trigger an autoimmune response in which the body immune system attacks its own tissue and organs. The resulting damage may be cumulative, such as occurs in examples are rheumatoid arthritis, multiple sclerosis, or acute, such as a complete destruction of pancreatic islets that can occur in diabetes 1. Some autoimmune disease in systemic lupus erythematosus, rheumatoid arthritis, Raynaud's syndrome, inflammatory myopathies. In organ specific, autoimmune atrophic gastritis of pernicious anemia, autoimmune hemolytic anemia, autoimmune thrombocytopenia, autoimmune hepatitis, Graves' disease, Hashimoto thyroiditis, multiple sclerosis, insulin dependent diabetes mellitus. Coming to hypersensitivity. Hypersensitivity is an immune response that damages the own body tissue. They are divided into 4 classes, type 1 to type 4 based on the mechanism. Type 1 hypersensitivity reaction is immediate or anaphylactic reaction, often occurred with allergy. Some symptoms can range from mild discomfort to death. Type 1 hypersensitivity is IgE mediated, released from mast cell and basophils. Coming to type 2 hypersensitivity. It occurs when antibodies bind to antigen on the patient's own cell, marking them for destruction. This is called antibody dependent hypersensitivity. It is IgG mediated and IgM mediated. First, type 1 hypersensitivity is allergic reaction IgE mediated. Type 2 hypersensitivity is antibody dependent hypersensitivity. It is mediated by IgG and IgM. Type 3 immune complex aggregation of antigen complement proteins IgG and IgM antibodies deposited in various tissue that trigger type 3 hypersensitivity reaction. Type 4 hypersensitivity means cell mediated or delayed hypersensitivity. Usually takes between 2 to 3 days to develop. The type 4 reaction is involved in many autoimmune and infectious diseases but may also involve contact dermatitis. It is the T cell mediated. Thanks for watching. Subscribe to my channel for more videos related